Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 32, going out on October the 24th. Among many other special days, it's World Polio Day. I shouldn't actually mention these special awareness days because when I don't have an interview related to it, which I don't, it feels like I've missed an opportunity. But don't worry, next week's will be just the same level of competence, or lack thereof. I think I've just about recovered from the 3 trips in 4 weeks, although I can't say I've made a massive amount of headway on editing. Is more podcasts the answer? Probably not. But those trips always leave a lasting impression. I really enjoyed Vilnius, as always, and Gothenburg for the first time, and Dublin for that matter. But this was a relatively uneventful week, which is probably just what I needed anyway. Last week we had a conversation with Biomatter Designs, and we also talked with Danu Sports, both of them from those previous trips. This week we're talking multi-omics with Lithuanian company Vugene, and we will discuss protein degradation, among other things, with Amphista Therapeutics. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us or sponsoring a podcast episode. So because it was not a hectic week this week, we're back on track, and so that means we can go through some of the news headlines you may have missed over the past week at deeptechdigest.com. The NLS Days winners were announced in Gothenburg, the FDA approved a lupus nephritis treatment, and improved cough detection technology has been developed for health monitoring. An eye treatment met its endpoints. New GenMab data show anti-tumor activity, and a resistant hypertension trial met its endpoint. We asked the question, is the thyroid the key to prostate cancer treatment? A low-cost vial could prevent wasted vaccines, and CN Bio launched an all-in-one organ-on-a-chip system. There were positive results from a Pfizer breast cancer trial. A genetic discovery offers hope for personalized prevention of cardiovascular disease, and you can read all of these and many more at deeptechdigest.com. Okay, first up this week is an interview about multi-omics data integration, among other things, from the Life Sciences Baltics event in Vilnius last month. I do miss that city. The audio isn't perfect on this one thanks to having to use 2 different recorders and some background noise that ranged from completely silent to extremely loud. Anyway, we talked with Vugene, a company based in Lithuania's second city, Kaunas. To learn more, we chatted with Gražina Mikelytieta, who is the CEO, but first with founder and CTO Juozas Gordevičius. All right, so first then, maybe could you give me a bit of background on the company?
So, Vugene is a bioinformatics or biomedical data science company, and we are taking care of our customers' data whenever they are running a preclinical trial or a later trial in multi-omics. So when people get genomics readouts, transcriptomics, epigenomics, metabolomics, proteomics, or in the best case where they have all of these readouts taken together. We know how to process the data, we know how to integrate and figure out what is the good statistically significant finding, what are the relationships between these multi-omic layers.
Where do you work with companies from?
Most of our customers are in the USA. Many are also in Europe, so USA and Europe are our primary markets.
All right, and everybody talks about the influence of AI. How has that impacted upon what you do?
Well, we love AI. We keep saying that we don't buy it, we produce it. And the reality is that many of our workflows are using AI, and part of the final result that we deliver to our customers are AI models that we evaluate, and we can tell them if we take this data, what will be its diagnostic accuracy, what could be the prognostic biomarkers that you can use for the treatment. So this is part of what we are doing, and we are very happy about this advancing technology, and there's bright future in it.
Does it matter what the data is? You can analyze results from anything?
We do focus on multi-omic data. So the trick is that many data scientists can analyze any data. We know what are the best industry standard methods, taking them from research, from academic papers. We evaluate the best methods and we apply those methods. So our customer does not have to be an expert in data analysis. Our customer has to be an expert in biology. They have to know what they are trying to achieve. We make sure that the data that they generated is actually showing everything that they were asking for.
And what are the benefits of working with a company like yours? Is it speed? Is it saving money? Is it the accuracy?
Speed is probably the main advantage. We say that we have multiple pillars that we are standing on. One of them is speed. The other one is reproducibility, as everything that we are doing is a piece of code, and that code can be traced back to who wrote it, when it was written, and every result can stand for itself. And finally, the AI part that we are able to build. So these things together make the main advantage of Vugene.
All right, so maybe tell me about the multi-omics.
One of the key innovations within our product is ability to study multiple omics and integrate across multiple omics layers. Traditionally, because of cost and because of the complexity, people would study only one layer of the omics processes in a cell. For example, gene expression, or only proteomics, or only metabolomics. Now, more and more people are realizing that you have to understand how these layers are associated one with another. How do those associations appear or break down in disease or during treatment. And that's how you understand completely novel mechanistic insights. And we have built this solution into our product that automates integration across multiple omics layers. And that leads people to very quick insights. So that also, for a researcher, that removes a pain point. If I will do an omic experiment where I'm not so familiar with the method, who is going to analyze my data? We'll take care of that. So it's much easier to do multi-omic experiments now.
And I guess that the more data that you have, the more those interconnected results can show things that just studying one aspect would not necessarily show.
Exactly, and there is also a very mathematical way to show that if you look into one layer only, or one layer here, and in another experiment you look into the other layer, there are things that you will not be able to see. Only when you look into them together and you see how they interact, you can look into these interactions, and in themselves they are revealing new mechanics of the way the cells are functioning.
And that's something that probably maybe even 10 years ago you wouldn't even be able to do all of that. You just wouldn't have the computational power.
It takes a lot of computational power, true, and of course leveraging existing models, leveraging the AI, leveraging existing data sets, all of that becomes easier and easier. So one more reason to be happy about the upcoming AI technologies.
Obviously you can't talk about individual companies because of confidentiality, but what kind of positive results have you been able to achieve for companies?
We have been successful in building good predictors of disease. We have been successful in identifying pathways and target genes that down the road led to new approaches, new experiments towards a new drug.
And how do you get clients? Do they come to you, or?
Most of our customers come to us by referral. So happy customers always bring new customers. And that's the main way that we are working. And of course, we are going to conferences, meeting people, making ourselves available, and looking for ways to increase our client base.
So could you tell me a little bit about the scene in Lithuania?
Right. So I think Lithuania is a very nice place to grow a business in general. There are a lot of young people, technologically savvy, biologically literate people who can join the team and help to grow the business. And since all our customers are in the US or in Europe, it's not too far away. So we are trying to be present also there, work with our customers. Many of them have said that they feel that we are collaborators, not doing fee for service, but we are actually there answering their questions almost immediately.
What do you see as being the future for your company?
Our company started as a service, but we have been able to productize a lot of the things that the way that we do them, we automate them. And even though many of our customers still don't feel that there is a product behind the scenes, actually the way we do things is very fast and very efficient. So we are able to scale it up immensely. And this is actually our goal, to make data analysis in biomedical sciences available immediately. Have you received your research data, your multi-omics data set? I wanted the insights from this data to be there with the researcher the next day, so that they can drive the next experiment, they can drive the next drug development immediately.
Do the companies that you work with already know a lot about the way that this is done, or do they just hand everything over to you and say, do it?
Usually, the customers hand us over the data and they just ask for the results. And of course, we explain every method that we have been using. For us, transparency is important, so we really show them what has been done, why the result is like this, why we suggest to make this or that decision based on the data that is there.
All right, let's turn it over now to have a few words with the company CEO.
My name is Gražina Mykolaitytė. I'm CEO of Vugene. I have joined Juozas to help to grow the company. And I think we have been successful over the last years, hiring really talented team, multidisciplinary team, with the studies finished at great European and US universities, and bringing all the talent and knowledge here to build our services and building the multi-omics data analysis platform. I think my biggest trust is the team, because we are the ones who are creating, which are delivering the best service to our customers, and which are able to develop the new ideas. I think what we were able to achieve a really great culture in the team. We follow the same values, and the biggest value that we create to the customer is that we are working together. We are cooperating, collaborating. We are looking for solutions together in the office, discussing the complex questions, and this is where really the biggest value arrives for our clients and partners and ourselves as well. As team is also willing to grow. They are willing to deliver the global level projects to work on the key scientific innovations. So this is actually a goal of mine and Juozas together, to be able to gather with the team to be on the top of the world with the innovation, with our product, and being able to deliver it to our customers on the global level. This is what drives us, the innovation, being able to deliver the best in class service, and grow together with all the market, help the life science industry to find out the treatments, help our scientists to find the best medicine for treatment of the people, the best solutions for people to live healthier. So that's also our vision, to help scientists to arrive to discoveries faster and with the higher quality. And for the future, we also aim to grow with the team as well. We have a very niche profession that we have, so we really need to find the good talents globally, not only in Lithuania, and this is where we will be probably growing in other countries, and bring the best talents to our team to follow our vision and to deliver the best services to the clients.
Maybe the same kind of question, but what is it about Lithuania that is it the standard of education? Is it the talent pool?
In Lithuania, we have multiple criteria helping to grow the business. First of all, really talented team and talents that we can find in the market with the really high education. Secondly, we have government institutions helping a lot from the first steps for the startups to grow and to scale. We have Invest Lithuania Innovation Agency, another agency is providing the credits and guarantees for the small companies with also VC funds helping the young companies to grow with their investments. So we really have a supporting ecosystem to grow from a small idea to the business and to delivering to the market. And I would say that all the life science ecosystem here is very helpful to each other. We have also Lithuania Bio Association, which helps to bring all the companies together, share the knowledge, encourage each other. We have bigger brothers and sisters which help us, which have advised us what to do, because we already made a few steps ahead, and they could give us the best advice to not to do the same mistakes that we have done. So I think that friendliness, talented pool, the government support, also the life science industry is one of the top priorities for Lithuania. So there is a lot of support here. And I think we are in the center of the Europe, so we are also very central to be able to support other European countries. And also from here, we can be global easily.
And I guess it's not just Vilnius, there's Kaunas as well, which has good universities.
Exactly. In Lithuania, we have universities in Vilnius, in Kaunas, and Klaipėda, three biggest cities where most of the talented young people finishes universities and then go to work for life science, biotech, and pharma companies here. Of course, Vilnius is probably the bigger hub than the other cities, but we're willing also to expand our system through all Lithuania.
If you needed proof of Lithuania's great life science companies, there's another one, and we still have more lined up for you from that event in the coming weeks. Next, we're talking about targeted protein degradation with Amphista Therapeutics and the company's Chief Scientific Officer, Louise Modis. Thanks so much for joining us today, Louise. I wonder if first you could tell us a bit about your background before we get to that of the company.
So I started my career working on transcription factors in myeloid cells, understanding how they helped cells decide what to do, whether they were going to proliferate or differentiation. So I studied that in normal hematopoiesis, and I studied it in AML. And it really captured my imagination back then that transcription factors and gene regulation was how you could get a cell to behave itself and how you could make cells do different things. And that has stayed with me over the course of my experience in drug discovery. And anytime I had the opportunity with various enzymes or receptors or ligands to hit in on a gene regulatory pathway, I found that really quite compelling. And so in thinking about my career, when I had the opportunity to join Amphista, that was in my mind. That was a big driver for me. Right, this is a technology where I can now get into the heart of the cell and tell it what to do by degrading certain proteins. So, and I think that's been a motivator for a lot of people in the TPD field. And certainly when I talk to my friends and colleagues across the field, they're just thrilled, as am I, by the idea of finally we can target these proteins we've known about for decades. We just couldn't get at them before. That is the promise from a scientific and therapeutic point of view that I see in the field that I find really quite compelling.
I wonder if you could tell me a little bit about the company.
Absolutely. So Amphista is a targeted protein degrader company based in the UK, in Cambridge, at the heart of structural biology, the home of structural biology, with a lot of great technology advances around So we have a nice small team all co-located here in our offices in Cambridge, developing new next generation targeted glues that can degrade proteins in a way that go beyond the first generation degraders. And we have a portfolio that we're really excited about and looking forward to talking to you more about that.
All right. So before we talk about what you do, then I wonder if you could set the scene a little bit with some background on what targeted protein degradation is and the relevance to that in treatments.
Absolutely. So targeted protein degradation is a really clever technology that allows us to do something we haven't been able to do historically. We can degrade proteins in a cell. And in addition, those molecules can be configured so they can be given as a pill. So this is a really big deal for patients and therapeutically has a lot of applications beyond oncology, where the field has started, into immunology now and has great potential for other diseases, rare diseases, CNS disorders as well. So degraders are really here to stay as a modality. They're really an established modality now. And this makes sense because therapeutically, this is what biologists like me have always wanted to do. We've wanted to get rid of the proteins that are doing something important in the cell, whether they are regulating gene expression, whether they are core proteins in a signaling node. That's what we want to do. And most of the time, we haven't been able to drug those particular proteins. So we end up, coming in other ways. We end up going after enzymes that regulate the pathways using kinase inhibitors, for example. Or we end up going all the way upstream in a cell to the exterior and targeting signaling molecules or receptors of antibodies. But when you give us the chance to actually degrade the proteins we want to degrade, it opens up a lot of new therapeutic space in oncology, but also in many other diseases, particularly immunology and CNS disorders. So if you think about it, if you want to degrade a protein that is at the core of the decision making in a cell, a degrader allows you to do that. The other thing is that sometimes proteins just accumulate. They just pile up, they aggregate, they accumulate, and you just want to get rid of them. And that's the other clear advantage of a degrader technology versus an inhibitor. An inhibitor isn't going to help you with excess of protein if it's got structural problems or structural implications. So really that's a therapeutic opportunity, and that's why we're so excited about it. What's great now is after 20 odd years of theory and preclinical practice, you now have dozens of molecules in the clinic degrading proteins. Largely, they're based around cereblon technology. One ligase out of 600 really dominates the space. And they've made just brilliant progress. And there's been an awful lot of money and time and energy put into getting from the concept, which was pioneered initially by Craig Cruz and his colleagues with Protax, through to demonstrating you could make these things work in animals, through to figuring out serendipitously that if you use cereblon as a hook, as a ligase, as a hook, that then could be used to tag the protein that you wanted to degrade. So this all came about, serendipitously, as almost everything we do in science does. There is a drug, lenalidomide, that's used as a cytotoxic agent in multiple myeloma. We knew it was leading to degradation of proteins, no idea why. It turned out it bound to cereblon. And that ligase then really stabilized interactions between other proteins and itself, tagged them for degradation, and took advantage of the cell's natural cleanup system, the ubiquitin proteasome pathway, which is just used to clear up, dead and malfunctioning proteins in the cell. And the ligases, as I said, there's about 600 of them. They're like your quality control managers of the system. They go around scanning the cell, looking for proteins that are damaged and weird in different ways, faulty proteins you might want to get rid of. And we just got lucky with cereblon, figuring out that was one we could use. And we've invested really, as a field, we've invested really heavily in that and made great progress with it. I mean, the field is established. Companies have sprung up left and right. There's a lot of money being poured into the space. And that then puts us in a parallel, if you like, to the antibody field historically. First, you show, can it work? Yes, you can make it work. Can you get into the clinic? Yes. Can you actually develop these things in a way that's safe and effective for patients and work with the regulatory authorities to get them approved? Yeah, you can pretty much. And now you look at how can you make them work really well? How can you optimize them? How can you go after other targets? How can you go into other space? And that's where the field is now. So there's a lot of energy around trying to find other ligases beyond cereblon or VHLs, a couple of the early stage ones. And there's a couple of reasons for that. The first reason is the ligands that you use to bind cereblon are, they're reasonably chunky. They have certain chemical features that we have worked with as a field, but they have certain liabilities that you want to get around. They have certain chemical features that make them a little awkward to work with. They're large, they have hydrogen bond donors. There's just things that are inherent to those molecules you can't really work around. So the first problem to solve for us is, can we make degraders that have better behaved chemistry? Can we make degraders that are more drug-like, easier to work with, and therefore can be taken into broader indications, either with better tissue distribution or better activity in different cell types, or just easier to configure as orally bioavailable molecules or CNS penetrant molecules. So that's the challenge for the field. People have gone hunting for new ligases to bind, and that turns out to be really tricky. And so at Amphista, we founded the company based on the idea of, we've got good chemistry that degrades, not really sure how. We've got good chemistry that degrades, looks drug-like, looks like something we can work with. So let's take a chemistry-centric approach and say, right, what makes a good-looking drug? Reasonable size, reasonable features. You want this thing to be potent, selective, orally bioavailable, manageable, manufacturable. So if you start with the chemistry at the center and see what you degrade, and then figure out which new ligases you co-opt, that could be a way to do it. So we kind of turn the paradigm around and put chemistry at the center. And over the last few years, we've done just that. We figured out that we actually, the way our chemistry works is, we don't pick the ligase. We don't say, this is the ligase we're going to use to degrade this particular target. We pick the protein we want to degrade. We choose a ligand for that protein. And then we add our chemistry to that ligand. And it turns out that the chemistry we add together with the ligand creates a new interface, creates a new PPI, new protein-protein interface, if you like. And that is recognized by the ligase that's best suited for that particular, to recognize that particular problem. So of all the 600 QC managers out there, one of them steps up and says, "This, I'll take this one." And we've seen that now happen in each of the programs we've worked on. So we have BRD9 going into the clinic next year with DCAF16. We have our SMARCA2. We have with DCAF16. We have our TEAD protein with FBOX22, and we've got a couple of others with other ligases. So we don't tell the cell which ligase to use. We say, hey, this protein looks damaged. And the cell says, right, and takes it out. And what's fascinating to us, and actually was a real pleasure as we worked through this over the last period of time, is that interaction is unique. When we knock out DCAF16 or FBOX22, we don't see any degradation of the particular target protein. So it's completely dependent on that particular ligase. So we get this really nice selective specific degradation, which was a very pleasant surprise when we did these experiments. It's really nice. And we think that's all linked to how we do our chemistry, how our targeted glues work. So we've coined the term targeted glues because we take molecules that bind to the protein of interest, a ligand, and that's our target. That's the target end of the molecule. And that's like heterobifunctional molecules historically bind the protein you want to drug. Then we attach our chemistry to it, and our chemistry is small and glue-like. And our chemistry has a covalent reversible warhead. When a ligase scans this complex, it recognizes, it binds transiently, we presume, and then this covalent tag, this covalent reversible warhead interacts with the specific cysteine on the ligase, for example, stabilizes that interaction long enough that the ligase can now tag and recruit the UPS system molecules and recruit ubiquitin and tag the protein for degradation. So when we get those initial hits, seeing that we see degradation in our system, and we don't know what ligase it is because we've done a phenotypic screen in cells, we've just looked for degradation of the protein of interest, that's our question. When we look closely and we generate cryo-EM structures, figure out which ligase it is, generate cryo-EM structures and look closely, we can see the interactions between that ligase and the protein of interest. We've really invested in cryo-EM ternary complex structures to get that information. With a chemistry-centric view, quite literally, with the VR goggles, the whole bit, our biologists, our biophysics, our computational chemist team sit together and figure out how they can make a molecule that really fits nicely at that interface between the two proteins. Building on the tag that we have through the covalent warhead, how can we create this nice glue interface and maintain the drug-like properties, of course? So we have our experienced, battle-worn chemists who've taken many drugs into the clinic, who know what they're looking for in terms of drug-like properties, participating in this process as well. So it's not an academic approach. It's very much a drug discovery, drug development guided approach. We build these molecules with the interface between the two proteins. So that's the glue-like component. They turn out to be incredibly selective. So when we do global proteomics in cells with those molecules, we degrade the protein of interest and pretty much nothing else. And that's really satisfying as well. So this for us is the real thrill of this space. So we're really pleased we didn't insist on picking one ligase and saying, right, we're going to figure out how to degrade using this particular ligase. We're really pleased that it's worked out, that the cell has picked ligases that work. Now, as additional stroke of luck would have it, the ligases that we've picked up so far are all really broadly expressed. So they're broadly expressed, they're consistently expressed. Our bioinformatics team here has scoured the literature and scoured the public databases, and indeed our internal data as well. And you see really nice broad expression of these ligases across all cell types and tissues, looking at various single cell databases. And if anything, they're upregulated in certain cancers in certain situations. The whole opportunity for targeted protein degradation going forward is, how can we make this work more broadly? How can we solve problems that exist with the first generation molecules? How can we pick up new targets and make it work in a more general way? So with our 3 new ligases in the mix, I'll tell you more about the next one on another podcast. Those ligases offer up new space. And with our team, we've a really tight team. We're all quite literally sitting here in the space in Cambridge together. I've built up this team over the last couple of years with a real focus on bringing diverse opinions and experiences to the table. So I kind of joke with the team, we have one of everything, but we really do. We have skills and expertise in a really multidimensional way. And then we draw, of course, on the powerhouse of Cambridge structural biology around the corner to get insights and ideas whenever it suits us. So that's worked really nicely for the team over the last couple of years. And so the timing now to share what we've been up to and to move our projects into the clinic is, it's just great. So it's good fun to be talking to you about this today.
The introduction of AI, has that accelerated things, or do you utilize that?
Oh yeah, absolutely. I should have said that more explicitly. So our structural biology team here, whether they're the cryo-EM guys, whether they're the modelers, whether they're the computational chemists, whether they're the design chemists or the synthetic chemists, they are all AI enabled. They are early adopters, innovators on the cutting edge of whatever new technologies are available to use. So we are absolutely using all of the available techniques, and indeed we're building our own databases and tools within our chemistry expertise to expand that. That really opens up the space for chemistry generally. Using AI, if you can figure out synthetic routes to make things, you can now have more imagination in what you design. If you can use cheminformatics and the energetics associated with structural biology knowledge to tell you what kind of shapes might fit, biology speak, but what kind of molecules might work, then you have the courage and the motivation and the energy to make them and to kind of fight through some tricky chemistry. And the general acceptance, growing acceptance of taking large small molecules into the clinic, it's really offered a renaissance of small molecule drug discovery. This field has really opened up the space. I've seen the chemists I'm working with now more excited than they've been for 15 years, thinking back on, when we were just working on GPCRs and kinases. They're just totally buzzed by the idea of what they can do chemically. And AI is a really big part of that. We, of course, use the obvious AI tools like AlphaFold and the related docking tools. But, the critical thing for making molecules that work is actually being able to see the protein interface. One of the things that has really helped us is having the cryo-EM structures at high resolution where we can see the interface between the proteins. We can see different docking configurations. We can see how the ligases shift and mold depending on the target. So we work through different cysteines using the same ligase, different cysteines on different targets. They really kind of warp around the target. So having the actual cryo-EM structure of your particular target protein interface is crucial. I don't believe that looking at a ligase in isolation you can figure out what it can bind to. You have to see how it looks. It's like Play-Doh. You have to see how it fits. And then when you see the shape of it, you see the mold, then you can do clever things and creative things afterwards. But being able to actually see what we're doing has helped us a lot.
You mentioned earlier other companies working in this space and your approach being I guess, almost a 180 degrees from what other companies are doing. Are there any other things that you're doing differently?
We have the benefit of having deep TPD experience based on the founders involved with Alessio and his team. We have the benefit of bringing together our team with really important expertise, if you like, across the arc. We're taking a really open-minded approach for what ligases are engaged. So that is the biggest thing. Being chemistry-centric first, being really open-minded about which ligases are engaged. They're really the critical things we're doing differently. We have the opportunity to get into the CNS, and that's huge. It's tricky. It's going to be a whole bunch of work, but that is huge. We've shown our molecules are CNS penetrant across a range of different projects, and that is inherent to the chemistry of our warheads. So I think that's the next big breakthrough for our company and for the field, is really being able to use these beyond oncology or beyond systemic applications. So I think that's going to be really important. And I think the next phase, as we think forward, where's TPD going to go? One of the big questions for drug developers generally is, how can I target cells in a more selective, more specific manner? And one way to do that might be to find ligases that are cell type specific or situation specific and drug them. We're not there yet as a field, but I don't doubt, given that the 600 ligases out there, it's just a matter of time to figure out which are these ligases that we can co-opt. So that's coming. The second thing is, can you attach degraders to antibodies and create degrader antibody conjugates? Just like you create cytotoxic agents attached to antibodies for the treatment of cancer, can you attach a degrader to an antibody to treat other diseases, to treat immunological diseases, to treat inflammatory disorders, to treat cardiometabolic diseases? In that case, you do want a more selective profile. You may not want to kill the cell, you may just want to make it do something else. And that's where a degrader can be used. And so there's quite a bit of energy in trying to generate degrader antibody conjugates. And we think, there's no reason why our chemistry can't be used in such configuration as well.
Would you see those as potentially working in tandem with antibody drug conjugates?
It'll probably depend on the indication. I think the antibody drug conjugates are brilliant for delivering a toxic payload to a cell in oncology, killing the cell, which is what you want. I think there are situations where you want to deliver a toxic payload to a cell in immunology as well. You might even want to kill the cell. And in that case, you're considerably more picky about that molecule leaching out and causing other effects, because you tend to take those therapies chronically for a very long time. I think that it's going to depend on the indication and on the target. So, I think there's combinations are... Biology is complex, so in order to cure the complex diseases, there's no doubt in my mind you have to come in either with orthogonal molecules in combinatorial fashions, and or go after the control center of a cell by taking out particular gene regulators or signaling regulators. So combinations, as long as they're safe, combinations will absolutely be picked up. It's just, you can see the march forward from oncology into HIV and into immunology. We are, as a community, we're getting more comfortable and more clever about how to combine things and doing things in series rather than in parallel. Different treatment paradigms are emerging. So yeah, absolutely, I would take nothing off the table when it comes to curing diseases.
Well, it's funny because I just thought about how we eat. You don't just have one diet. You have so many different things that go up to make what your diet consists of. And it would seem that it would be an exactly similar approach in treating things, is to use as many different strategies as you can to tackle all the various aspects that a disease exhibits.
Yeah, absolutely. And the brilliant thing is, every time we take a therapy into the clinic and we think it works in a certain way, we learn so much about how it works once we actually take it into the clinic. Meaning we learn so much about the therapy, but we also learn so much about the disease. We're brilliant at coming up with great hypotheses and fantastic models for things. But when you take a molecule into the clinic and see what the disease does in response, that tells you what your next move should be. That tells you what your next therapy should be. That tells you what your next target should be. I mean, it is fabulous to have watched things like different medicines and how they've taught us about disease. And we end up, re-stratifying diseases and regrouping patients on the back of how a particular medicine works. And that I think the targeted protein degradation field is absolutely going to enter that space as well, as we see different degraders in signaling pathways being taken into the clinic. I just can't wait to see what they're going to teach us about those diseases and then where we go next. So it's an absolutely brilliant time to be a biologist in the therapeutic space, having tools like targeted protein degradation and antibody drug conjugates coming through. It's so many different things we can think about now that weren't possible 20 years ago.
And you recently announced some data from, is it SMARCA2?
Yeah, so we've had a couple of press releases recently, one on SMARCA2, and we're pleased with that. And the important thing about that is we've got really nice, potent, selective degraders that are selective over SMARCA4. And as trivial as it is to say that, it's actually really difficult to do that. SMARCA2 and 4 are virtually identical. And without our cryo-EM structures, where we could really see the interface between SMARCA2 and DCAF16, and we could see how SMARCA4 bound, without that knowledge, we wouldn't have been able to kick SMARCA4 out of the way, if you like, and to get those nice degraders for SMARCA2. So we're really excited about that because that's what's needed in the field. SMARCA4, if you degrade too much of SMARCA4, you have toxic side effects. That's been seen in various early trials. And you really want to keep that SMARCA4 degradation to a minimum. And we believe we have a profile that has the potential to be best in class there. We can also see evidence of oral bioavailability in our chemistry. We can see evidence of CNS penetrance in our chemistry. So that opens up therapeutic possibilities, long-term therapeutic possibilities for SMARCA, because SMARCA2 is for the treatment of lung cancer. And it turns out in about 10% of lung cancers, SMARCA4 is mutated. And SMARCA2 is left behind to carry the load all by itself. So when you degrade SMARCA2 as well, those cells, those lung cancer cells, are expected to die. And it's what they call a synthetic lethality hypothesis. You need either one or the other to keep cells alive. If you take out both, the cells will die. And that's why you want your SMARCA2 degrader to be really selective over SMARCA4. You want the cancer cells to die. They've already lost their original SMARCA4. You don't want the neighboring cells to die. So you can't take out SMARCA4 as well, or you'll just have too much toxicity. So taking a degrader like SMARCA2 into the non-small cell lung carcinoma population has generated a lot of energy and a lot of interesting deals in the field. It has the potential to be really transformative for those patients. And those patients, people already, those patients get profiled. You know what their karyotype is, their genotype is when they come in for treatment. So you know who to treat as well. So it's got a lot of potential. So we're really excited about that one.
How does what you're doing relate to TEAD? And could you tell me about how that works and what it's being utilized for?
Absolutely, yeah. So TEAD is another protein that is part of a complex that isn't traditionally, it's not an enzyme, it's not traditionally accessible with small molecules. However, a number of groups have figured out how to block a protein-protein interaction between TEAD and YAP1 and to block the signaling downstream of that molecule, also for the treatment of lung cancer, starting off with mesothelioma and then has the potential to be effective in lung cancers beyond that. So what's interesting about that approach is that they block a specific protein-protein interaction, but they don't take TEAD out of play. So presumably other protein-protein interactions still exist. And of course, the protein is still there. So we looked at this and we thought, I wonder if you degraded TEAD, would you get better efficacy? Would you get deeper pathway inhibition? Would you get broader pathway inhibition? And so we tested that. So this is an example of, cancer is a tricky beast. Sometimes you need to take out every last molecule in the signaling cascade to get the effect you want. So that was our logic behind that one. And we did indeed make degraders that are highly potent, highly selective, really fast degraders. So even like we do, when we dose them in vivo, we see complete degradation within 2 hours. So really fast degraders, and our molecules are already bioavailable. So that's great. What's interesting there is that deep, fast degradation translates into really deep inhibition of the associated biomarkers. So we think there's an opportunity for this to have greater efficacy, broader efficacy, more sustained efficacy than just blocking the protein-protein interaction alone. I think that comes back to how our targeted glues work. Again, we have the cryo-EM structures there. We can see the interactions. And I think by making a targeted glue that just mirrors this interface beautifully and really makes this nice tight interaction. It's energetically favorable for the ligase to bind the protein. It's energetically favorable. It just works. It just fits nicely. And you just don't have any delay. You don't have any forcing. You don't have any dragging of a ligase that doesn't want anything to do with the target. You're just not forcing it in. You're just allowing it to bind nice and tight, little super glue operation, and then the protein gets degraded. And we think that that's our best guess for what's responsible for the speed of the degradation that we see. It's really it's the super glue analogy. You've broken pieces of porcelain and you just glue them together nice and seamlessly, and it works nicely.
The only problem with that is it sticks to your fingers and you can't get it off.
It does stick to your fingers, but maybe super glue isn't the very best example. It's more like a piece of parafilm, a long sticky interface between two proteins that so some small weaker interactions across the whole interface are actually together giving you the affinity to bind. And we see that when we look at our structures, we see lots of small weak interactions. We see various, the guys are modeling the various water groups and all sorts of energetics from all sorts of angles, and taking advantage of all these little interactions to have a nice binding across the interface. So the molecules can, they're a reasonable size and they can span a decent area. So you pick up this interaction across a long interface.
As far as your company's concerned, what does the future hold there? What's your strategy in terms of where do you go? Is it clinical trials? Is it working with other companies? What's the end game, I guess?
Yeah, absolutely. So we are taking BRD9 into the clinic for AML next year, and we've a good-looking molecule who does the kinds of things we want, fast, potent degrader, all that good stuff, orally bioavailable. And we also have done a bunch of work on the biology behind that, and so we've come up with some novel insights into how to use that drug in the clinic, and we think it's going to be transformational in AML and be able to use it across a broad range of patients. We'll talk about that data at the ASH meeting in December. That's a key activity for us for 2026. We want to take our earlier stage programs forward into the clinic, TEED and SMARCA, of course. We already have collaborations with other pharma partners, for example, with Merck. And our technology is great. But there's only 55 of us. We have to stay focused. We've got to get the business done on these molecules. So we're absolutely open to collaborations with others who have specific things they would like to work on, particularly in the CNS area where, you want to engage with people who have deep expertise in CNS targets to take advantage of our chemistry and our honed techniques on the chemistry and structural biology. So I see those as the main activities over the next period of time.
It is very difficult, well, not difficult necessarily, but to not overstretch yourself and go into too many different directions at once.
That has been the challenge. And when I joined the team, certainly, we have, diverse backgrounds between us. We said, right, we just have to focus on a couple of projects, one indication where we let's just be really laser focused on these couple of projects. And that's really, over the last couple of years, that's really paid off. It's obviously hurts a bit to say, right, we're going to park all of these fabulous ideas for the foreseeable future. But, the team have rolled with it, and we've really made nice progress on the couple of projects where we're all in on. But yeah, we have the blinkers on all the time, and it's not that we don't want to do other stuff. We don't have other ideas. We indulge them ever so briefly, and then we get back to work on the couple of projects where we need to make progress. So yeah, that's the critical thing. But I think that's true in any organization. If you don't go all in on the projects you care about and have the whole team all in on it, it's hard to win even when you are all in. So we really push that laser focus.
You've already mentioned earlier about where you see the field could go, I guess, in terms of the biology and the chemistry of things. Where do you think that it could potentially go in terms of what it can do for ultimately the end user, i.e. patients?
Yeah. So I think that the steps forward now are not conceptual. We know we can degrade things. We know we can scale them up. We know we can work with regulators. We know we can get them into the clinic. And so those practical drug discovery problems are solved or being solved or will be solved. So that's covered. From a patient point of view, if I was a patient, I want to be able to take this thing in a pill, not 3 times a day. Thank you very much. I'd like to take it, somewhat intermittently. I might, once a day is fine, once a week would be even better. Having degraders that are orally bioavailable, that are safe. I don't want any weird rashes or any things happening to me when I take these things, and have long-lived effects. So having, extending their half-life in vivo, for example. Choosing targets that don't turn over so rapidly. So the nice thing about going after gene regulatory proteins, for example, is they're not typically post-translationally modified, or they are typically post-translationally modified rather than transcriptionally modified. If you degrade the protein, you often have a couple of days before it comes back, and you can shift the cell state. So I do have a view that we will be able to, in some indications, get in, shift the cell state, re-establish homeostasis of a system, whatever that system is, and that you don't have to take this thing chronically, perhaps. I mean, you can take it for a period of time, reset your system, and not have to take it chronically or not have to take it round the clock. So I think that kind of patient experience of taking something intermittently, taking it in series with other medicines, I see that is where the field is going to go. And that goes hand in hand with understanding how disease evolves, and goes hand in hand with the advances we're having in understanding diseases. All the deep and large data sets we're generating about disease at a single cell level and at a patient population level, learning about how a disease evolves and thinking about it as kind of having a personality that you have to shift in different ways. I see that as how our treatments to disease is going to be in the end. I mean, it comes back to the early days when I started working in biotech. It was all about personalized medicine when I was back at Millennium. And that's still the dream. That's still where we're going. It's not quite personalized yet, but we're getting there bit by bit. And I think that's the opportunity for TPD as well as other technologies to offer the patients a personalized approach to their disease as opposed to the disease that they happen to have.
And it seems to be that it's not just a finite field that you have what you're working on now. I think there are infinitely more possibilities as you move forward, which must be a nice field to be in, that it's not kind of rigid.
Yeah, it is. It's great. I mean, it is the idea of in the fullness of time being able to develop degrader antibody conjugates, being able to find ligases that have more restricted expression profiles or situations where they act more specifically. When we pick up those ligases, that's going to open up more space. The idea that we can get some of these medicines into the brain. I mean, the fact that our chemistry can get into the brain, as long as we don't attach a ligand to it that has all sorts of properties that won't work. But, being able to get into the brain, the idea of being able to target things in the CNS is super thrilling for neuroinflammation, which is more my background, for neurodegeneration, where we're aware of lots of different aggregated proteins there where you just want to get rid of them. That is a really thrilling area. And it's really stimulating when you go to conferences or meetings or read the literature. The kinds of things that people are coming up with is fantastic. And that's, not even touching on all the different LITACs and other kind of ways of degrading extracellular proteins and membrane proteins. So it's a real renaissance in the chemistry, but it's also a renaissance for the biologists. We get to go, right, what were the targets I have been wanting to target for the last 25 years? And you can smile to yourself and say, is there any sort of a ligand that can bind to this thing? And as long as you can get, for our targeted glue technology, as long as you can get a ligand that binds to that protein, sort of, at a decent level, it doesn't have to be brilliant, it doesn't have to be in any particular place, as long as you can just get it to bind. It doesn't have to be selective either, because we can engineer in the selectivity with our cryo-EM structures and our ternary complex information. You now have a hook, you have a starting point. We take the approach of just degrading directly in cells. We're not theorizing too much about it. We're saying, right, let's see if our chemistry can degrade this target in a cell. And if it does, yes, we'll dig in. If it doesn't, we say, thank you very much, next target. We don't try to insist. We say, if it doesn't work, in a cell with a couple of goes, let's just move on. Because we have a decent sized library that we screen with a bunch of different reactive warheads. We feel like we sample the space pretty nicely. So we're taking a phenotypic approach, a phenotypic screening approach to finding targets. But it really is, we have the biologists and the chemists are excited at the same time. And that's always a really good place to be, because people then are sparking with each other and And I really enjoy that. I enjoy bringing people together and fomenting chaos briefly and getting people to argue and to negotiate and to think about things and just think beyond what we know to be true from our past and think, what could we do? Like, if this is true, what could we do? And so we have a nice mix of really experienced drug developers and some much earlier career scientists who are not bogged down by knowing what didn't work in the past. So that environment is really fun to work in.
You don't want to put the cart before the horse necessarily, but it's exciting that in a lot of cases, especially with things like neurodegenerative diseases, what we're talking about now is slowing things down to extend life a little bit in some cases. But what you're talking about is the potential maybe at some point for even reversing some of these conditions. And you can't necessarily call it a cure, but if somebody's not having their disease develop and it goes the opposite way, then that's just incredible potential.
Yeah, it really is incredible potential. We know diseases take absolutely ages to develop. If you can slow them down, if you can give if you can take these pathogenic proteins out of play in certain systems, our system, we're remarkably able to regenerate ourselves. We have incredible capacity to problem solve within our own bodies. And many organs have the capacity to kind of adapt, if not regenerate, but really adapt and take that opportunity for the healthy cells to really take over. We're pretty well buffered as a system. So it does really offer the opportunity to, the nice term in cancer, it's you're living with the disease. You're not dying for it. You'll die with it. You won't die from it, kind of thing. And that's where we want to get to with all of the diseases. If we can get to a point where people have a chronic condition that they keep under control, that they manage with a pill once a day or once a week, I think almost everybody can live with that. And the cardiovascular world has taught us, with some exceptions, you get people who take their blood pressure medication and their statins, and they're living along happily, and their lives are extended, and they literally don't even know they're sick because they're not sick. They just happen to have a condition that you're now managing. And if we can get there with autoinflammatory diseases and neurodegenerative diseases and metabolic diseases, that's just brilliant. And we are chipping away at it. We've made just incredible progress in therapeutics generally, and TPD is definitely a new and really established modality at this stage. We're just waiting now for the fruits of that to show up and have drugs that we can actually give to patients and go, here, have a degrader for this, that's going to sort you out. Take it every day now, or whatever the instruction happens to be.
Yeah, and I know you were mentioning about once a week, but I think if you had the opportunity to stop something in its tracks, you'd take 10 a day if you needed to.
You know what? You would. I would. Yeah, you would. You'd. That's for really serious diseases. Compliance isn't an issue, and we have some really serious diseases we haven't been able to make much inroads in. And I think for us, tackling diseases like AML, where very little has changed really since I was working on FLT3 inhibitors 20 years ago, we really need to move the needle there. And, something like BRD9 may be able to move the needle there. And it's taking out a protein that you just couldn't drug before. So that kind of thing, like, can we move the needle on diseases like AML? Can we move the needle in terms of lifespan on diseases like non-small cell lung carcinoma? We can imagine, and this is known in the field, combining SMARCA and TEAD with each other, combining SMARCA and or TEAD with PD-1 antagonists, combining them with standard of care. That's going to be the way it rolls. If it can extend people's lives with a good, decent quality of life, that would be fabulous. They're the kind of things, we don't want to make Me Too stuff or kind of iterate. The kind of things that attract me and the team I'm working with is to do something that really makes a difference, makes a splash, feels like it was really worth it,
Next week might have to be one of those bumper editions with an interview from Lithuania, Sweden, and our regular guests. I guess it's a good problem to have. Too many interviews is better than none at all. Regardless, Amphista is definitely a company to watch, and that was a really interesting conversation as well. Anyway, that's it for this week's podcast. If you do want to get in touch with guest opportunities, which lots of people are doing, or if you want to advertise, please do feel free to contact us at jim@deeptechdigest.com. I think it would be nice to have a quiet weekend for a change, but it's looking like it will be dry. So I expect we'll be hitting the trails while we still have some good daylight hours. It's also that time of year when countries are resetting clocks, although you never know which ones and which way. I generally mess up interview meetings at this time of year by getting the time difference hopelessly wrong. So if I am interviewing you this week, please join the call either an hour early or an hour late. So until next week, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next week for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.